welcome everyone to our 2026 Know All Your GBM and Recurrent GBM Treatment Options Patient Disease Educational Webinar. I'm Delanne Elliott Midland, and so I am your chairman and CEO of the End Brain Cancer Initiative, formerly the Chris Elliott Fund. And I'm proud to be here moderating this particular webinar with each and every one of you today. Right now, we are going to have Dr. Lilly speak to us about CSF liquid biopsy for GBM and recurrent GBM. Dr. Lilly is amazing.
Thank you, Delanne.
He earned his PhD in molecular virology and was then introduced to the challenges of rare diseases and cancers during his postdoctoral research on oncolytic virotherapy. Okay, what is oncolytic virotherapy?
Oncolytic virotherapy is basically taking viruses and then genetically engineering them to kill cancer cells, but to leave normal cells intact.
Wow.
So this is something that has actually been explored in brain cancers. I was doing it in multiple myeloma, but it is a very unique repurposing of pathogens to be able to turn them around and make them become therapy.
Ooh, that's exciting. We all need to learn more about that too. I think we'll have to do a webinar on that.
Awesome.
From his postdoc, he entered industry to leverage his passion for strong science and science communication in a sales role at Cell Signaling Technology, where he developed his commercial acumen. From there, the clear patient need led him to joining the liquid biopsy space, first as a scientific affair liaison, then as an MSL, medical science liaison, for cell-based and cell-free technologies for monitoring and characterizing cancer. He brings his 5+ years of experience in liquid biopsy and interfacing in the healthcare space with healthcare providers and key opinion leaders to the team at Belay Diagnostics, where he has joined as MSL, Medical Science Liaison, to bring his passion for cutting-edge and patient-centered medical science to the challenging field of central nervous system tumors. It is my pleasure to introduce Dr. Lilly, but before I do, there's 3 takeaways he wants you to take away from today's talk. And the first one is CFS testing with Belay Diagnostics can inform diagnosis of GBM and recurrent GBM without surgery or biopsy. Just think about that. Secondly, Belay's testing covers all guideline-indicated targets for GBM and recurrent GBM to inform treatment. He'll talk more about that. That is very, very intriguing alone. Repeat testing is now possible using Belay's testing to monitor disease evolution. So this is a tool. This is an amazing tool that a lot of people really don't know about yet for GBM and recurrent GBM. Dr. Lilly, we are excited to welcome you to the stage and to educate us about Belay's Diagnostics.
Thank you so much. It is always a pleasure to be here with EBCI and to speak to such a passionate and incredible group of people about something which I am very passionate, and that is how can we use the things that we've learned in science and the methods that we've developed to make a real impact on how patient care is actually progressing, especially in extremely challenging areas like glioblastoma and recurrent glioblastoma. This is something that has touched my life and friends of mine, and so I have a little bit of a particular vendetta against this devastating disease. And so it is a pleasure to be here speaking on behalf of my company, Belay Diagnostics, to talk about how we can utilize the fluid that cushions and protects your central nervous system, your brain and your spine, which is called cerebrospinal fluid, or CSF, to actually detect the genetics and genomics of cancer, in particular in glioblastoma and rGBM, and that's what's called liquid biopsy. So I'll give you a little introduction about who we are at Belay and why we're here and why we partner with brain cancer. Well, the big thing about us is that we are tried and true, dyed in the wool for the purpose of central nervous system cancers. Our co-founder is Dr. Jathan Bettigauda. He's the chair of neurosurgery at Johns Hopkins. And the testing that we provide is built on more than a decade of research and development in his lab and in the lab of his mentors. So this was something that, even though we are a young startup company, is built on well over a decade of established science and tremendous data suggesting how useful this kind of technology can be. Our assay was built on the shoulders of that great research, specifically for utility in cerebrospinal fluid for central nervous system cancers. This is not a test that was originally developed for some other kind of specimen, so it's not a tissue test, it's not a blood test that was then readapted for use in this arena. It was purpose-built. And that gives us a real edge, and it is part of why our testing is so accurate and why it performs so exceptionally well. Something that we are very, very passionate about is that we are very much a science-minded company. We want to collaborate with researchers and clinicians, and we also want to make sure that whenever we make a claim about our testing and how well it performs, that that is something that can be checked. So all of the validation studies that back up the claims that I'll be showing you today are peer-reviewed and published scientific manuscripts. This is something that we believe very strongly in because the validation should be transparent and it needs to be relevant. So it needs to be validated in diseases that are important and relevant for the kinds of patients that are going to be tested. And it needs to be something that both providers and passionate patients and caregivers can look into. One of those validation studies in particular is the validation study for our flagship product, which is called Summit 2.0. We talked a little bit about comprehensive genomic profiling. Our test is one of those. And we were able to do our validation study using CSF samples that were also matched to a tissue biopsy result. So we were able to get a really clear performance comparison against what is really the gold standard for getting profiling of cancers in the space for GBM and for rGBM is still tissue. It's what they take out of you when they do your resection. We're in the unique and wonderful position to be able to offer a path forward to that same kind of information in instances where surgery may not be possible. So just a little timeline of us here. We were officially founded in January of 2022, and we came to the market last year as a fully fledged commercial entity in January. We launched the first version of our test, which was called Summit, and its companion product Vantage, which is important for looking at one particular biomarker that's actually important in this family of cancers. And we learned very quickly that the offering that we had was good, but it wasn't enough for clinicians in this space. And so in less than a 10-month period, our lab and the incredible people that work down there were able to turn around a full 2.0 version of this test that increased the number of genes that we looked at by more than 10 times, added a number of other biomarkers, including those fusions that Dr. Wong mentioned. Those are a part of our testing now as well. I mentioned that we're a very science-minded company. We like to publish, and our publication, of course, is not just our validation studies looking at both, the performance at the molecular level, but how well they provide the correct clinical information, but also published case reports. We have 5 published case reports out now. There are several more coming. And one study I did want to highlight a little bit is a study that was recently published, and this was a 120-patient single institution study at UT Southwestern. And the goal of this was to understand if the testing that we were offering was having a meaningful clinical impact on the care of those patients. And the short version of that is that in 93% of the cases, and this was even using our more limited Summit 1.0 test, in 93% of cases, our testing was important for helping to have a meaningful impact on managing care for patients. And this impact came in the form of helping to inform diagnosis and guide treatment, to confirm the response to therapy, and even in some of the cases that were negative in that study that came back with no cancer-associated genetic variation or no mutations, they helped to aid make the correct diagnosis in that patient. And so this is one of the ways in which our testing is really helping to shape the future of medicine for central nervous system cancers. And this is something that is very near and dear to us and is a huge motivator for us, is to make sure that we are really doing everything that we can for patients and those who care for them. This will be the last of the slides where I talk about us as a company, and then we'll get more into talking about the tools and how they apply to you. In the time that we have been available on the market, we now have worked with probably over 170 now institutions. And the majority of NCCN designated cancer centers are regularly performing this test on patients with central nervous system cancers from across the entire spectrum of disease. These can be both primary central nervous system cancers that arise from brain tissues like glioblastoma, or from metastatic brain disease, things that metastasize to the CNS exist, and we play in that space as well. So our tool is designed to give that information in a way that's very, very important for helping move care forward. And so you can see the institutions that have deployed Belay Diagnostics testing. This is, of course, a non-exhaustive list, but if you look across here, you'll see centers from across the United States of all sizes and shapes. We work also in the pediatric space as well. And so our goal is really to help this field mature and grow into the next era of being able to use really excellent precision tools to help manage patient care.
Cameron, this is Delanne. I have a question.
Shoot.
On Belay's website, do you have all 160 plus institutions on there that are ordering Belay's tests?
That's a fabulous question. We don't, and the reason we don't is because some institutions don't want us trading on their name, and they're very protective of their identity. However, if there are patients who are curious about where to get our testing, they can call customer service. You may end up speaking directly with me. The other thing you can do is you can speak to your neuro-oncologist and have them call us, and we can work out how to work with the doctors you're already seeing as well. So we are here to help you have access to our tests. It's not just, we're here and it's in these institutions and that's it. No, we are interested in trying to make sure that this is available for the patients that need it. So I would say start up a conversation with us or have your doctor do it. We're here to help.
Awesome.
Great question, Delanne. Okay. So we're going to back way up. We're going to take a 10,000-foot view at glioblastoma. So what are we actually talking about when we say GBM? This is a particularly, let's call it insidious brain tumor. Glioblastoma is aggressive and it tends to invade the tissue around it along the nerve fibers. And so it's very difficult to get it all out when you go in for a resection, and it grows very fast. The invasive nature of it is part of what gives it this very high recurrence rate. So even in cases where you have successful therapy and you have successful surgery with good margins, you got a good gross total resection, there are still cells that are in there and eventually they will come back. This particular tumor is part of a family of brain tumors that arise in the cells which care for and protect your neurons. These cells are called glial cells. The family is called gliomas, and glioblastoma is categorized as a grade 4 astrocytoma. So the cells that give rise to that are astrocytes. These are important for kind of cleaning up after neurons, feeding them and protecting them. But when they start to divide out of control, they can cause cancers like this. Glioblastoma is the most common malignant adult brain tumor. In the population, brain tumor is still a relatively rare disease, 3 cases per 100,000 people per year. But with that said, GBM accounts for about 15% of the diagnoses of new brain cancer patients every year. So that's still a number of people every year in the United States and across the world that now learn that they're living with an extraordinarily challenging disease. The diagnostic pathway for this. So how do you go from I'm not feeling very well to, oh, I have a confirmed diagnosis for glioblastoma, is something that I don't think I need to belabor to the audience of this call particularly much because you've lived it. It starts at the same place most CNS disease starts, and that's with imaging, so with an MRI. Next, they may do some testing on your cerebrospinal fluid, which is the arena where we at Belay play, they may perform what's called cytology, where they spin out the cells using a centrifuge and then look at them under the microscope to determine if there are cancerous cells present. This has a pretty low sensitivity. It's not the greatest test. But just recently, and by just recently, I mean about 3 weeks ago, the treatment guidelines for all gliomas, glioblastoma included, now include a recommendation to look at tumor-derived DNA in CSF to be able to do this kind of genetic characterization utilizing CSF if surgery is infeasible. And when we say infeasible, this may be because the lesion is in a location that is inaccessible. Trying to access that via surgery would cause massive cognitive problems or other medical issues, and so it's not possible. But it may be that the patient just decides that they don't want brain surgery. So surgery refusal is a real thing. But now there's an option here, and this is something that you may not have heard of because this technology is still very new. And the guidelines just updated on April 24th to reflect this new application. And we think that this is a great portent for how tools like ours, but especially how our tool, because I will say I do think it is the best one available, can actually be employed to help care in this space move forward. So again, as I don't think I need to tell too many of the rest of you, the next step is almost always surgery. If they can do it, they'll go in for a maximal safe resection, get as much of the tumor out as possible. They'll take out as much as they can. But if it's not possible to go in or they really need to get that diagnostic confirmation, they'll go in for a stereotactic biopsy. And then from there, they look at exactly what Dr. Wong was talking about to look at the nucleic acid from the tumors and do a comprehensive genomic profile. And that looks at a number of different genes for numbers and numbers of different variants. And especially in the recurrent space, this can be performed to look at targets and options for therapy. And that's something where we, on our side of things, come in. Because these challenges are very real in diagnosing and characterizing glioblastoma in real patients. MRI is not specific. You can't necessarily always say that it's a glioblastoma from MRI. Other central nervous system cancers and some other central nervous system diseases that are not cancer can look like cancer. CSF cytology is a standard of care measure, but it's very limited sensitivity, and it has a high, what we call false negative rate, whereas the test comes back negative, but the patient is eventually diagnosed to have cancer. So that's not great. One other big challenge, as I mentioned, is that resection and biopsy, these are invasive procedures, and they're not feasible in every case for every patient. And definitively diagnosing glioblastoma basically requires some kind of genomic testing. So we have to have access to be able to distinguish glioblastoma from other gliomas that might look the same on MRI and might cause the same kinds of symptoms. So this is a thing that needs to be performed and is a part of how this is designated to be worked up by the treatment guidelines. And so our testing does basically the same kind of thing that they're doing on tissue, but they do it using cerebral spinal fluid, which we can obtain via a spinal tap. So it's much less invasive, and it gives us the ability to look at disease in much the same way. Our testing is designed to be used across the entire journey for a patient. At the time of diagnosis, it can be used to help guide and indicate therapies. It can be used to detect responses to therapies. We can track basically the level of the amount of nucleic acid which has a mutation in it compared to the total amount, and we could look at that proportion. And if treatment is being effective, we should see that number come down. That allows us to assess response. And it can also be used as a part of surveillance and monitoring to detect those molecular signatures for recurrence, sometimes even before MRI may be able to, because MRI does have limits on the size of things it can detect. When we talk about comprehensive genomic profiling from testing with Belay, we're talking about our two tests, which are Summit 2.0, which is our large flagship multi-gene panel test, which looks at a number of different biomarkers, but also one that we talk about is called Ascent. This looks at sort of the structure of your chromosomes, and it looks to see if arms of chromosomes are missing or extra arms are there, or if small regions of those chromosomes, but that are still much bigger than individual single genes, are present in more or fewer copies than they should be. This is really actually important in glioblastoma, as glioblastoma has some very, what we would call canonical or commonly found alterations at the chromosomal level. And so Ascend can even aid in the diagnosis by just finding those. Vantage looks at methylation of a gene, which is called MGMT, and methylation of the promoter of this gene tells clinicians that your disease is likely to be sensitive to temozolomide, which is something I believe a lot of you are probably going to be familiar with. And so this can actually give you that full comprehensive profiling out of one single CSF sample. And that's another thing that's very important about what we do and the technology that we have licensed from Hopkins is that all of this can be done on 6 ml, which is something like half a teaspoon of cerebrospinal fluid. So let's talk at a very high level about some of the content of the test that we have, and then I'll walk you through some actual real-world patient cases in which our testing was utilized to understand GBM in the clinic, in the wild. So I mentioned Summit 2.0. This is our comprehensive genomic profile testing in CSF. It is the most comprehensive testing available, and that's based on the number of genes that we target. We look at a very large 520 multi-gene panel. The importance of this, again, is to be able to get at those, what we like to refer to as molecular insights or molecular characterizations, and that's looking at what mutations are present or not. It helps us to understand the exact genetics of your specific cancer, and that helps care teams plan treatment, understand risk, understand prognosis, and make those diagnoses correctly. We are validated against tissue biopsy, and those results are peer-reviewed and published. Again, as you can tell, we're very proud of that. One thing that's very important for us, and especially in this space, is that our turnaround time for results on a CSF test are 10 to 14 calendar days, not business days. And that's really important to us because we understand that there's a patient on the other side of that sample. That CSF belongs to a human being that is suffering from something that is changing their life in a major way right now. And we want to get the information into the hands of the care team very quickly. Our validation study allowed us to find what's called the clinical sensitivity and specificity. Did we detect cancer in the appropriate samples? And did we get the right kind of information that we were supposed to? A clinical sensitivity of 96% and a clinical specificity of 98% is industry leading. No one else out there that is looking at trying to do the same kind of testing, their tests do not perform anywhere near this well. And we are really dedicated to making sure that we provide the best tests. And so we're very proud of how this performs. I mentioned that this very large panel testing looks at 520 total genes. These genes include all of the genes that are important and recommended for testing in glioblastoma and recurrent glioblastoma. We look at what's called copy number variation for 62 of those genes. And so that looks at, are there extra copies or fewer copies of a given gene than are there supposed to be? Some of these, especially in glioblastoma or a recurrent glioblastoma, one of those is called EGFR, and that's on our panel as well. Dr. Wong introduced the concept of gene fusions and how those can have an impact on glioblastoma and the trajectory it can take. We look at fusions for 28 target genes, including the ones that he mentioned in NTRK and in BRAF, but 26 other genes. And that can help us inform, treatment and understanding the diseases and moves on. The next two that we look at are biomarkers. One, TMB, is short for what's called tumor mutational burden. And that looks at basically, if you look at 1 million base pairs of DNA, how many times do you see a mutation? High tumor mutational burden has direct implications for particular kinds of therapy. And so if we can detect high TMB in a patient, they have, an option for therapy that they otherwise wouldn't have had without that result. Another one of these measures is called microsatellite instability, and that looks at basically how much the genome is behaving correctly. are the very ends of the chromosomes the right length or not? And that can also help understand, especially things like metastatic breast cancer. So it's a little bit less relevant for GBM, but this breadth of our panel is part of what gives us the ability to say conclusively that we have the most comprehensive genomic profiling that you can perform in cerebrospinal fluid. So I mentioned testing for these larger, because we think about a chromosome as being very, very small, they're microscopic, but a chromosome has tens of thousands of genes on it. And so when we're talking about the kinds of things that can lead to the loss of a chromosome arm or an extra copy of one, those are what we would call pretty large genetic insults. And we can look at those both happening at the arm level, but also at small regions of the chromosomes that contain a few genes or important genes to understand if those are present in extra copies. The validation study for this was actually just recently published. It's one of our more recent publications. And we were also able to determine a very, very high sensitivity for both different kinds of chromosomal alterations that we can see and a very high specificity as well. We had no false negatives in this particular assay. And I did mention that this is designed to pair with Summit 2.0. They are designed to work hand in hand with one another. Another thing that's very important about this particular test is that you don't need a lot of nucleic acid. And so in some patients, there isn't a lot of the tumor DNA and the tumor nucleic acid that's shed into the cerebrospinal fluid. So sometimes a scent can come back positive and give meaningful information where there actually isn't enough nucleic acid for a scent to be successfully performed. So this is a very important pairing with Summit 2.0 to get at all of the information about a given patient's specific cancer. And lastly, I mentioned Vantage. This is, of course, for looking at the methylation of the MGMT promoter, which predicts sensitivity to temozolomide, which is called Temodar on the brand level. This is, again, something that is highly sensitive and highly specific. All of our tests can be performed on their own, but they are really designed to work with one another. And all three of our tests can be run on the same 6 ml sample of cerebrospinal fluid. There are some very special things about our methodology that allows them to be done so. And one of those is that we use a different actual technique inside the test tube itself to determine the methylation status here. The canonical way is a chemical method, and it ends up being very harsh and it destroys about half of the DNA that's put into it. We use an enzyme that does not destroy half of the material that's put into it. And so because we retain that precious template, it gives us the ability to do this on very little input. And so again, that's part of why we're able to perform so well and do this all on one sample. So let's talk a little bit about from a very high level. I will not just hammer you guys with a list of gene names because we would be here forever. But Summit 2.0 contains all of the guideline recommended genes and biomarkers for diagnosing glioblastoma and to plan therapy looking at targeted application of specific kinds of treatment in the instance of recurrence. This is something that is very intentional. We wanted our test to be able to be utilized in this way in glioblastoma. And so at any time during your care and your journey, you can get a snapshot of where the disease might be in its evolution with this test. As I mentioned, a scent can detect chromosome alterations. Well, in glioblastoma, this is a disease that has a very, very common pattern of some chromosome arm level alterations. You get a gain on chromosome 7 and a loss on chromosome 10. This is seen in something like 3/4 of glioblastomas and is diagnostic in and of itself, especially if all of the other markers point to a glioma. That is a characteristic signal for glioblastoma. I mentioned about Vantage and how that can be utilized for temozolomide sensitivity. And one thing that I definitely wanted to point out is that our reports not only give you, one, the actual results, what was found, what mutations. The comments section also includes some interpretation of those, but they also list clinical trial options on the report based on the results from that specific sample. in the relevant kinds of disease. And so if you are looking for a trial, that's a great place to start because you can take our report to your care provider and say, how do I get on this trial? Is this a good trial fit for me? And one of the other big things for us is that because this can be done without the need for literal brain surgery, and you can do this using a spinal tap, you can actually track the disease over time. You can monitor a patient and see how their GBM is evolving from your initial testing and look at it over time. And we will put all of those results on a nice graph so you can actually look at how this is going for you when you get our results. So let's talk about 3 case studies in which our testing was employed in the arena of patients with glioblastoma. The first of these cases describes a situation in which our testing was employed to try to get at that diagnosis. There was a 61-year-old male patient who had some concerning symptoms, had gone in for an MRI, and they found a new lesion. So this was concerning for at least a glioma, if not a high-grade glioma, of which GBM is one. But the location of this patient's mass was infeasible for surgery and biopsy. And so the consideration here was, how do we actually get at the correct information to characterize the patient's tumor? So the care team in this case decided to run all 3 of our assays, Summit 2.0, Ascent, and Vantage. And you can actually see on the right side of the slide, this is a de-identified copy of this patient's actual results. So you can see what our results will look like. To a clinician, this may mean a lot or a little. One of the other things that we do is we provide results reviews with clinicians as well. I will sit down with the doctor and walk them through the report and help them understand it. But what we found was a number of mutations that were characteristic for and helped to inform the diagnosis of glioblastoma in this patient. So we found a mutation in the promoter region for a gene called TERT. We found multiple copies beyond the normal number for EGFR. This is something that is also quite canonical in glioblastoma. And we did find that chromosome 7 gain and chromosome 10 loss that I mentioned before as being kind of characteristic for GBM. We found a few other mutations as well. This is one of the interesting things about running a large multi-gene panel, is that sometimes we get surprising results, and results that are teaching us things about the disease that we didn't have access to before. For example, this particular patient actually had an ERBB2 mutation. And so for those of you who may not know, that's HER2, which has immense applications for and importance for characterizing and treating breast cancer. And so we're learning through tests like ours that that's right, Mandy. Absolutely. You mentioned at the start of our call, Mandy has GBM and breast cancer. Oh my goodness, Mandy. But so you can see that this gives you, we're able to discern a tremendous amount of genomic information that can allow a care team to plan not only, one, the immediate diagnosis, determine treatment, and may give them information for how to address the disease even when it recurs, if it recurs down the road. And it also gives, at the time of diagnosis, this is a fantastic baseline that now at the time of recurrence, they can go back to compare to and see how the disease has changed. And that can help patients, and that can help caregivers, and that can help providers keep applying the best medicine for that specific patient. This was a very interesting case. It ended up being a new diagnosis of GBM, but the initial tissue biopsy was inconclusive. And so this is another arena in which our testing can be very useful, is if the biopsy results come back and for whatever reason you get a non-diagnostic biopsy. There is a small rate of that does happen. And I can't imagine the frustration of having undergone a stereotactic biopsy to then not get the diagnosis afterward. So this is an instance where our testing can be very, very powerful. And so this 64-year-old female patient was highly suspect to have a glioblastoma based on the imaging results, based on the symptoms. but the initial biopsy did not give enough information to be able to make the diagnosis. And so Belay's testing was performed in this case to really try to resolve that particular diagnostic uncertainty. And what we found were both two mutations in and many extra copies of EGFR, along with some variants in some other genes which have been known to be present in glioblastoma. And so this was enough for the care team to go, all right, so we had a non-diagnostic biopsy. Our CSF, comprehensive genomic profiling, gave us the information we needed to try to get to that information. But it wasn't just that. It was that we were also able to turn around information about the tumor mutational burden in this particular patient too. As I mentioned, high tumor mutational burden is an on-label indication for what's called immune checkpoint inhibitors. And so if you've ever heard of PD-1 or PD-L1, I don't know, maybe something that you've heard on TV or seen in a commercial, these are things that can really help patients with TMB high disease. And so a TMB high, in our case, is enough to be on label for the application of that therapy. So now not only do we have genetic variant information, gene amplification information, we've now been like, okay, and now there's a treatment avenue here. When we looked at this patient's chromosomal pattern of alterations, we again found that really canonical chromosome 7 gain, chromosome 10 loss. But one of the other things that I think this does a great way of highlighting how the looking at focal alterations could be, so alterations in small areas of chromosomes, is we were able to detect copy number losses, so fewer copies than normal, of two other genes that are very important for making this diagnosis, or three, excuse me, CDKN, 2A and 2B, and a gene called MTAP. You do not need to worry about what any of those are, aside from that detecting those was helpful in resolving this diagnostic problem. And they ran Vantage on this patient, and we found that the promoter was methylated, indicating this patient would be sensitive to TMZ. So they went from non-diagnostic biopsy to informing the diagnosis and having options for therapy right off the rip with 1 6 ml sample of cerebrospinal fluid. They didn't even have to have brain surgery. And then the last case I'd like to talk with you guys about is looking at recurrent glioblastoma. Recurrence is a slightly different challenge than the initial diagnosis, because primarily on initial diagnosis, anybody that can will get a resection and temozolomide, or they'll get temozolomide and radiation, or all three. Recurrence is a little bit of a different animal. And one of the things that's recommended in recurrence is to have this kind of comprehensive genomic profiling to look for novel targets for therapy. But it can also be utilized in this particular case to see if the disease has changed or grown. And so in this 69-year-old female patient who had a history of GBM that had already recurred once before, there was concern for what's called leptomeningeal spread. What does that mean? So the meninges are basically the membrane that wraps your brain and your spinal cord. They are responsible for keeping it healthy. They're actually where the cerebrospinal fluid lives, is inside some of the layers of the meninges, and spread to that is not uncommon in some kinds of cancer, GBM included. You can actually get the cancer trying to live in that space, effectively in the cerebrospinal fluid itself. And that comes with a whole host of other challenging symptoms, and it's difficult to treat. This patient had had a broad array of previous therapy. They had undergone resection, temozolomide, and radiation. They were also on tumor treatment fields at the time of the concern for recurrence and spread. The resected tissue from this patient, they had actually gotten genomic profiling on that, and this information was provided to us by this patient's care team, so we were able to compare what was found previously to what we found. Previously, they had found a mutation in the gene EGFR, which you've heard a lot about from to me today, but there were also fusions and extra copies found of EGFR. They had a variant in the promoter for the TERT gene, which I talked about a little bit, copy number loss of two genes, P10 and then CDKN2A and 2B. And when they assayed the tissue from this patient, the promoter for MGMT was found to be unmethylated, which would indicate a less good sensitivity to temozolomide. Methylated means sensitive, unmethylated, less sensitive. So at this point, the care team wanted to utilize Belay's testing to, one, characterize the disease and to determine if it was actually progressing. And so we found some different things than the tissue had found. And this is actually due to some underlying biological differences that mean that some things are detectable in CSF only. And it also gives you an ability to take that real-time snapshot. And that's what we found. So we did find the TERT promoter variant, but we did not find the other mutations that had been initially found on the patient's first resection. This indicates that the disease is evolving. But one of the other big things that we found is that at this point, that patient's MGMT promoter was methylated. So whereas before they had not really had a high sensitivity to temozolomide despite being put on it, now the methylation of that means that they will likely continue to be sensitive to it. Now, as we always say, and especially as I like to say, this always requires what we call clinical correlation. Your doctor will take this information and wrap it up with the sum of the information that they have from all of their testing. But we were talking with Dr. Wong about new tools and utilizing tools. This is a new tool in the toolkit, and it can be used in some instances, especially when you can't get to it surgically, to get the necessary information for diagnosis and informing treatment. And so I'll wrap up. One, CSF profiling using our testing can help make a diagnosis in this case in arenas when surgery is not possible. And it can be a great option for monitoring disease as well, because you can't really do surgery one after another, after another, after another, after another. Whereas a lumbar puncture, if you could potentially do that as frequently as every few weeks. In cases for suspected progression, our testing is very powerful. It can both determine that what is happening is indeed likely to be recurrence, and it can also provide therapeutic targets in the instance of recurrence and also reveal disease evolution. Cancer is nasty. It does not like to be predictable. And so retesting and understanding the genetics and genomics at the time of recurrence is very important. And this is a really fantastic suite of tools to be able to do that. And something that thinks a little bit more future state is that molecular characterization. And so when I say that, it's the kind of testing that we need. So if you hear that term, molecular characterization, we're talking about looking at nucleic acid, DNA, RNA, looking for mutations, getting an understanding of the subcellular biology that's going on. Molecular characterization of GBM using CSF liquid biopsy from us is extremely powerful. And this is a really excellent tool for the emerging era of managing central nervous system cancers and GBMs as well. And so I hope that this has been informative. I hope that you've learned a lot. I hope that you now know something about a tool which is out there and available on the market today. And with that, I'd like to thank you very much for the opportunity and the honor to come and speak to you. And if you have additional questions, I invite you to contact us. You can always reach out to customer service at contact@belaydiagnostics.com or give us a call. Our CS representatives are wonderful. They work in the office just a few rows over from me at our headquarters in Chicago. And my particular personal information will be available as well, so you're always welcome to reach out to me. And so with that, thank you so much. And what questions do we have?
Thank you, Dr. Lilly. Amazing presentation and amazing progress in diagnosis of GBM and recurrent GBM, but also, like you said, I think about the tools, right? I think about, literally being on top of this disease and knowing before you can see it on an MRI that if there is progression, you see it within the testing. This is an interesting question. Would this liquid biopsy serve as a replacement of molecular testing if you do not have any more good tumor tissue left for testing? I think you answered it, but could you be really specific.
Yeah, quite potentially. If you say have had a biopsy or a resection and that tissue has been exhausted in looking at other markers, our CSF testing can absolutely be used to get testing without the need for additional tissue collection.
Okay.
Absolutely.
And could this be done at any time, even after treatment has begun?
Yes, it can. And in fact, utilizing our testing at any time, you don't have to have this at the time of diagnosis. And in fact, oftentimes we're brought in to help understand the disease over time once treatment has been initiated, because the therapy is being successful. We should see basically the amount of those variants, what we call the variant allele frequency, which is expressed as a percentage. We would see that go from a larger number to a smaller number, or sometimes disappear entirely. And so it
Uh,
is never a wrong time to begin applying this kind of testing to get that longitudinal monitoring.
Excellent. And then, good question. Do clinical trials accept this test?
Fantastic question. The answer is unfortunately a very definitive, it depends. There are some trials for which they absolutely require what they will usually call histopathologic confirmation. And that means they took tissue, put it on a slide, and looked at it and were like, yes, diagnostically, based on this tissue, it's X. Some trials do not. And so that's something that you and your doctor should investigate, because there are trials out there that will take our data as enrollment criteria for clinical trials.
I would say, too, as more and more people utilize these diagnostics, there will be more information, there will be more data, and it takes a while, but we will probably likely start to see more and more and more clinical trials accept your tests
That would
as,
be the hope. That'd be
yeah, I mean, that's what I expect to see happen. And if you need any advocacy help on that, we are here. So there's lots of questions, something about insurance.
ah.
Um, yes.
So coverage right now is something that we're actively pursuing. We have started submitting claims for our testing to private payers. We've had some success with that. The big challenge right now is that we're out of network with most payers, and that means you're hitting high out of network deductibles. What I will say is that we also will work with you. If you
Oh,
want to get our testing, our company has extensive financial aid available. We have a fabulous, fabulous access and claims individual who can take your insurance information, run it with you right on the phone. They'll ask you a couple of questions about your other financial burden and tell you what insurance would charge you. We can tell you what we can do for you on financial aid. And in some institutions, it's not an issue because we have what's called a lab service agreement where we work straight with the hospital and it's billed the same way, a blood test might be billed. And it's a complete non-issue because that's billed as in-network. So
okay.
there are
Awesome.
options there. We're working on increasing our private payer coverage. We're also going to be going after Medicare coverage as well. That's something that we want, because one big thing is we do not want to add extra financial toxicity to patients that are already dealing with cancer.
Yes. The In-Brain Cancer Initiative would be happy to provide a testimony letter for Medicare, Medicaid. We'd also be very happy to testify when that time comes, because we know once that is approved, it filters down to individual insurance companies.
Yes, it does. Well,
Yes.
we will keep you in mind for that. I'll make sure that when the time comes, we'll connect you with our market access specialists for that exact. That'd be wonderful.
Fabulous.
Cameron, there was a question that Karen had asked. So she was wondering about if this test could help sort out if you are seeing real progression or treatment effect on an MRI.
Yeah, so it absolutely can. The molecular signatures of progression versus treatment impact, or what's called pseudo-progression, are very different. And that can be something that you see as a result of radiation therapy, among other things. But the utilization of our testing can help distinguish between both progression and pseudoprogression, and also response and pseudoregression. And that's one of the real benefits of being able to look at CNS disease using cerebrospinal fluid as a sample type for the kind of profiling that we do.
Thank you. And then she also had a follow-up question. She has the clinical trial options given on the report. Are they specific or in general terms?
So those are going to be specific to what kinds of variants were detected on your Belay test. So they're custom tailored from multiple sources, including clinicaltrials.gov, to be relevant to the patient for whom the report is about.
Gotcha. Thank you.
You're very welcome.
That's very helpful. Very helpful. Thank you. Thank you, Dr. Lilly. I think a lot of the questions were answered, not all of them. Again, you see on your screen, contact@belaydiagnostics.com for any questions you may have.